Morning everyone. I am Leena Prabhakar Jogi. I am teaching you a basic of phytochemistry, that is the unit 5 in semester 5th. So there are some contents of basics of phytochemistry. In that, first one is the modern method of extraction, second one is the applications of the latest techniques like spectroscopy, chromatography and electrophoresis in the isolation, purification and identifications of the crude drug. In next slide, there is the definition of extraction. What do you mean by extraction? It is the method of removing active constituents from a solid or a liquid by means of liquid solvents. It can be defined as the process of isolations of the soluble materials from an insoluble residue by treatment with the solvent. It is done for the separations of the medicinally active portions of a plant or animal tissues from the inactive or inert component by using the selective solvents. In this method, the wanted components are dissolved by the use of selective solvents known as the menstruum, and undissolved part is a mark. After the extraction, unwanted matter is removed. Then there are three major concepts in the extraction. First one is the extract, that is the preparations of the crude drug which contain all the constituents which are soluble in the solvents. Second one is the mark, that is the solid residue which is obtained after extraction. And third one is the menstruum, that is the solvent which is used for the extraction. Next slide is the related to the extraction principle. So extraction is the process of efficiently dissolving and separating the desired constituents from the crude drug with the use of solvents. It is controlled by the mass transfer. Plant constituents are usually contained inside the cells, therefore the solvents used for the extraction must diffuse into the cells to dissolve the desired components, whereupon the solution must pass the cell wall in the opposite directions and mix with the surrounding liquids. An equilibrium is established between the solute inside the cell and the solvent surrounding the fragmented plant tissues. The choice of solvents depending upon the characteristics of the secondary metabolites like polarity, pH and thermal stability. Next point is related to the list of solvents which is useful in the extraction. So there are some list of the solvent like acetic acid with their polarity as well as boiling point in the degree Celsius. So there are some solvents like acetic acid, acetones, acetonitrile, benzene and butanol, carbon tetrachloride, chloroform, cyclohexane, dichloromethane, dimethylformamide, dimethyl sulfoxide, dioxane, ethanol, ethyl acetate, diethyl ether, heptane, hexane, methanol, pentane, then petroleum ether, pyridine and propanol, tetrahydrofuran, toluene, water and xylene. So these are list of some solvents which is used in the extraction process, which is totally depends on the polarity of the drug. So like dissolve like is the principle on which the extraction will be carried out. Their next point is the ideal properties of the solvents. So ideal property, it should be solvent should be highly selective to the compound to be extracted. Then second point is it should not react with the extracted compounds or with the other compounds in the plant material. It have a low price, that means low cost. Then it should be harmless to the man as well as environment. It should be completely volatile. Then it should not mix with the water. It should not have it should have the big capacity in relation to the extractive. Then the density of the solvent should be different from the water density and should have the minimum viscosity. Next is the choice of extraction method. Choice means factor which is based on the extraction. So first one is the sample size, second one is the quantity of the extract required, third one is the extraction time, fourth one is the choice of solvent, and fifth one is the cost. Next, drying of the crude drug. So this is the first step in the extraction process. It is necessary to avoid the microbial contamination, as we know that if there will be the some water content is present in the drug, then there will be the chances of the microbial attack or microbial contamination. So we have to avoid this situation. So we have to dry that particular drug or crude drug so that there will be the essential step in the extraction. So it is done after the collection and before the drying. It will be done after collection as well as before drying. Then it is unless specified, drug should be dried below 60 degrees Celsius. Then there are different types of the drying. First one is the shade drying, then second one is the sun drying, third one is the far infrared drying, fourth one is the vacuum drying, and fifth one is the oven or hot air drying. In first shade drying, there will be the shade can be used for the drying purpose. That means it is useful for the drug which is highly volatile containing drug. That means which requires less exposure to the heat. Then it also there will be the less chances of the chemical alteration in the shade drying. Second one is the sun drying. It is most efficient and economical method. It does not require any types of the equipments or apparatus, so it is so costly or cheap method. It is use less intense sunlight. Then third one is the far infrared drying. In this less explored it is costly because of using highly expensive drugs as well as instrumentation is highly expensive. Fourth one is the vacuum drying, in which low pressure applied as well as rapid drying method, and it is suitable for the thermolabile compounds. That means the compounds or the drugs which are having heat sensitive characteristics, so this drug can be dry by using vacuum drying. And fifth one is the oven or hot air drying, which is used often. Next slide related to the mechanism of action, mechanism of extraction. So there are three main images you can see here. In that first image shows that the green colored plant cell, and in blue color which indicates the solvents. So in this stage, in this first condition, the solvent get penetrated into the plant cell by the action, that is the diffusion. So in second step, second figure, we can see that the solvent get penetrated into the drug and the solvent dissolve whole active constituent into that solvent. In third step, we can see that the drug get converted into the solution and move toward outside, that means into the solvent. So this is the mechanism of extraction called as the mass transfer. It is called as the mass transfer because in this situation or in this mechanism we can see that the drug active constituents which is present inside the drug get converted into solution and get moved towards the solvent. So this is the mechanism of action. Next is the step of extraction. In this there are certain steps, 5 steps in the extraction method. First one is the size reduction. As we know, there are different size reduction parameters, like we have to reduce the size of that particular drug to get the maximum effect or maximum extraction. So it maximizes the surface area. So as we know that less particle size, that means higher surface area. So as well as surface area get increases, there will be the increase in the mass transfer. Then next point is it is 30 to 40 mesh size is optimum. Next one is the extraction process, in which maceration, percolation, oxidation, these are the different kinds of extraction is there by which we can extract the particular active chemical constituents from the drug. Next one is the filtration. We can filtrate the extract by using masculine cloths, filter papers as well as filter press. Next step in the extraction is the concentration. What do you mean by concentration? That means evaporations of the extra solvent which is present in the extract. So we does not require the extra solvent, so we have to evaporate that solvent. So by using different aids like hot air oven, vacuum chamber, then rotary vacuum evaporator can be used to concentrate that particular extract. Next step is the drying, in which spray drying can be used or spray dryer can be used for the drying purpose. Next is the maceration. There are different techniques of the extraction, in which there are some conventional techniques like maceration, percolation, infusion, digestions, and some are modern techniques like Soxhlet apparatus. Then second one is the vacuum. Uh, so what do you mean by maceration? Maceration, that means soaking. The whole or coarsely powdered crude drug is placed in a stoppered container with the solvent, which is allowed to stand at a room temperature for a period of at least 3 days with the frequent agitation until the soluble matter get dissolved. So the mixture then is strained. The mark, that means the damp solid material is pressed, that means the mark is pressed. The combined liquid are clarified by the aid like filtration or decantation after standing. This method is best suitable for use in case of the thermolabile drug. In this slide we can see the jar which containing the drug, that means our crude drug with solvent, and you can see that there will be the change in the color of the solvent. Due to what? Due to extraction purpose. There will be the extraction will be followed up, and there will be the change in the color of the solvent. So in this process solid ingredients are placed in a stopper container with the whole of the solvent and allowed to stand for a period of at least 3 days, that is 3 to 7 days is the criteria for maceration with the frequent agitation until the soluble matter get dissolved. The mixture is then strained through the sieve or the nets, the mark pressed, and the combined liquid clarified, that means cleaned by the filtration or by decantation after standing. This is again the image or figure in which we can see the maceration process. This is the tree diagram of the maceration, in which plant material which is crushed or cut small or moderately coarse powder get converted into the coarse powder. Then it is placed in a closed vessel. Then menstruum can be added. Then it allowed to stand for 7 days and shaking occasionally. Liquid gets strained off. The solid residue, that means mark pressed, recover as much as occluded. Then strained and expressed, the liquid mixed, then again clarified by the substances and filtration, and then again evaporations as well as concentration. Then there are different types of the maceration, in which first one is the simple maceration. It is used for the organized as well as unorganized crude drug. For example, tincture of the orange, tincture of the lemon, tincture of the squill. These are the examples of the simple maceration. Second one is the double maceration, in which concentrated infusion of the orange is the example. Then third one is the triple maceration, in which the maceration process may be carried out with the help of heat or stirring. For example, concentrated infusion of the cassia, concentrated infusion of the senna. Next slide is related to the organized as well as unorganized drug maceration. So maceration of the organized drug, that means drug plus entire volume of the menstruum, and maceration of the unorganized drug, in which drug can be extracted with 4/5 portions of the menstruum. Then in maceration of the organized drug, the drug can be shake for 7 days. In maceration with the unorganized drug, shake the drug with occasionally on the day, that is 2 or 7. Then strain liquid and press the mark. In the maceration of unorganized drug, decant the liquid and mark is not pressed. Then third point is the mix the liquid and clarify by the filtration. Final volume is not adjusted. For example, vinegar of the squill, oxymol of the squill, tincture of the orange, tincture of the lemon, and tincture of the skill. In unorganized drug, there will be the third point will be the filter the liquid and final volume is adjusted by remaining menstruum. Examples are the compound tinctures of benzoin, tinctures of myrrh, and tinctures of tolu. Third one is the modification to the maceration, in which we have to modify the maceration process by using different aids. In that, it is used for the unorganized drug like gums as well as resin. So pressing is avoided in this modification to maceration, and fresh menstruum can be used to wash the mark as well as make the volume. Then next point is the multiple maceration, in which to obtain the concentrated extract, total menstruum divided into two parts, that means double maceration, or three parts, that means triple maceration, and each part is used for the maceration separately. The next one is the vacuum maceration. When pressure is reduced to desired extent, menstruum is allowed to enter, that means increases the permeability of cell wall as well as reduces the extraction time. Next one is the kinetic maceration, in which the vessel is agitated in constant motion for first 24 hours, then 20 to 50% more yield usually, and remaceration, that means first maceration carried out with the small amount of the menstruum, that is followed by the remaining amount of the solvent, and circulation maceration, that means solvent is continuously circulated by the help of pump. Next one is the percolation.